A study that was just released yesterday looked at the effects of childhood stress and positive parenting on development of the hippocampus and amygdala. I'd like to talk about that, and I'd also like to look at a previous study that examined the effect of epigenetics on activity in the amygdala in response to environmental stress. I'm Cameron, and this is my channel. Welcome to the Sexy Universe. This image from ResearchGate is the Coritolimbic system, which consists of the hippocampus, amygdala, anterior cingulate cortex, and dorsolateral prefrontal cortex. And this system impacts ability to focus, how we regulate emotion, how we form memories, and how we react to stress, so things like anxiety and panic, as well as motivation. This study looked at how childhood stress and parenting affect the growth of those structures. And specifically, they looked at the size of the amygdala and the hippocampus. There's one on each side, as you can see. Past research has shown that children who experience physical abuse and neglect tend to have smaller hippocampuses, but it doesn't appear to have any impact on the size of the amygdala. And other studies have shown that positive parenting behaviors, which they describe as warm, validating, and responsive, allow that hippocampus to grow. And they note that folks with that positive parenting tend to have better cognitive control, emotion, and they make better risk-reward decisions. They also note that folks who didn't necessarily receive that positive parenting did tend to benefit from youth interventions to help them learn emotional regulation. And as far as stressful events in childhood, they didn't just look at things like that happened within the family. They also looked at things like bullying and neighborhood violence. The study looked at 482 kids between the ages of 10 and 17. The average age was 13 and 4 months, and the proportion of male to female was 60-40. When you look at the income distribution, you'll notice that more than half come from relatively well-off households. That's kind of common in research, unfortunately. It's harder to get information on low-income folks. When they looked at childhood stressful experiences, they looked at how the participants experienced it, not how other people reported it. And they used a 0 to 4 scale. And basically, they looked at negative and positive events, how they felt, and whether they seemed to get better or worse. So these charts show behavioral problems as they relate to childhood stress and positive parenting. So basically, the less positive parenting, the more behavioral problems children seem to have. This chart shows the size of the hippocampus and how it relates to childhood stress and positive parenting. So more positive parenting tended to have a larger hippocampus, and less positive parenting tended to result in a smaller hippocampus. It didn't seem to matter how parents and caregivers reported these experiences. It was how the children perceived it. And the main benefit of positive parenting was that it created a stable environment where the kids could learn social skills and emotional regulation. So there's a huge positive impact of giving people a chance to retell their stories. That's the end of part 1 of this video. If you're enjoying the content so far, please click the like button and leave any thoughts or questions you have in the comments section, because it helps the YouTube algorithm. In the next part, I talk about a study that was done on marmosets, how they reacted to stressful situations, and how that related to epigenetics in their amygdala. In my last video, I talked about a study that looked at the relationship between childhood stress, parenting, and the development of the amygdala and hippocampus. And what the researchers found was that children who had a lot of stress growing up tended to have smaller hippocampuses, but the structure of the amygdala seemed to be unchanged. It's pretty well known that environmental stress affects the functioning of the amygdala and the prefrontal cortex along with the anterior cingulate cortex. But how does it do that? Well, I think this study can help shed some light on it. Now, the caveat is this study was done on marmosets, not on human beings, and I think you'll see why. The study involved 3 sets of experiments. First, they did a human intruder test in which they put the marmosets one at a time into a room like this, and they had a person stare at them for 2 minutes straight, which I don't know about you, but that would probably make me anxious. Well, they tracked the behaviors of these marmosets as they were stared at, and they gave a different anxiety score to each of these behaviors. Moving around and sitting at the front actually reduced the anxiety score, while these others raised it. They broke the monkeys they tested into two groups. One group they euthanized, and the other they saved for later. The marmosets that were euthanized, they tested these sections of their brain. And what they did, they took the tissues and they looked for specific active genes. And specifically, they looked at the epigenetics on these, and they found that this particular gene was activated in the right ventrolateral prefrontal cortex and the right amygdala. And the right ventrolateral prefrontal cortex would be that red part up there. And it turned out that the marmosets that had the biggest fear response had the most activated copies of that gene. So it's in the epigenetics. And just to talk about that for a second, there's a chromosome. The DNA is all wound up in that chromosome. So this isn't actual DNA, it's a rope, but maybe we can use it as a little bit of an analog. And it's all kind of held together with methyl groups that act like clips. So when a gene gets activated, first proteins in the nucleus take off the clip, and then they unwind it onto a rod. And once it's on that rod, it can be read and used by the rest of the cell. Well, if this gene that's been activated doesn't get deactivated later, it continues to produce a fear response. And yes, that epigenetic component can also be passed from parents to offspring. So when we talk about things like generational trauma, there is an epigenetic component to it. So one good question is, what can we do if those areas of our brain are kind of running wild? Well, studies show that one thing that affects them all is breathing. So learning to control your breathing can help. The truth is, environmental stress affects all of us, and it's going to affect both the structures in our brain and the structure of our epigenome. I want to talk about a few things science says we can do to improve those aspects of ourselves. I always have to give the caveat that I'm not a medical professional or a mental health professional. I'm just saying what the science shows. If you need medical advice, please consult with a doctor, nurse practitioner, or other medical professional who can give you the information you need. I will, of course, include links to these studies in the description. This first study looked at the epigenetic effects of exercise on skeletal muscle. This study examined 8 males who were basically living a sedentary lifestyle. All of them were aged from 19 to 27 years old. And they took part in 5 spin classes a week that each lasted for 60 minutes. And they did this for 6 weeks. Samples were collected from the participants both before and after the training, and it was found that 176 genes were downregulated by the training, and 641 were upregulated. The researchers found that several of the genes that were upregulated, that were activated by the exercise program, were also related to decreased risk of cardiovascular disease, healthier blood lipid profiles, and improved kidney function. This study looked at the effect of nutrition on epigenetics. And this is a meta-study, so it's a collection and examination of several other studies. They found that 2 nutrients that are closely tied to epigenetic repair are B12 and folate, because both of these nutrients donate methyl groups that are then used to turn off genes that shouldn't be active. They also mention other compounds such as sulforaphane, which is found in broccoli, and resveratrol, which is found in grape skins. And the paper lists several nutrients and food sources and how they affect the epigenome. Again, talk to your doctor or a certified nutritionist before you embark on any large dietary change. The authors also noted that the diets of parents can affect gene expression in their offspring. And they include this cool little infographic that compares the Western diet with the Mediterranean diet. The final thing I want to talk about is meditation and its effects on the epigenome. The researchers looked at 5 different types of meditation: mindfulness, Vipassana, yoga, Tai Chi, and quadratomotor training. And this also was a meta-study that looked at the results of other studies and evaluated them and put them together in one place. The researchers note that folks who practice yoga and Tai Chi tend to see improvements in stress responses, depression, overall mood, but the exact pathways aren't really known. they don't really comment on the effects of these practices on the epigenome. They note that quadrato motor training is a form of movement meditation, and they also do not comment on its effects on the epigenome. However, they note that QMT has been shown to improve reflectivity and creativity by stimulating neuroplasticity. Neuroplasticity is the ability of the brain to adapt to new situations. Vipassana is a sitting meditation, and the effects of this on the epigenome have been examined. And the authors note that in one study, participants who had 10 years or more of experience had their epigenetic profiles compared with people who didn't have much experience. And it was found that there were 64 different methylated regions, so regions that had been closed up and put away, that corresponded to 43 genes involved in glucose homeostasis, lipid metabolism, protein folding, neurotransmission, and modulation of inflammatory pathways. And when you talk about something like protein folding and inflammation, those are things that affect the entire body. They also took a look at a study that examined mindfulness meditation that was used to treat folks with PTSD. And they found that there were two genes, SLC6A4 and FKBP5, that had both been methylated. They had been downregulated or partially deactivated. And these two genes are potential biomarkers for depression. And in their conclusion, the authors note, and I'll just read directly, surprisingly, meditation practices seem to act on the same gene targets, such as FKBP5, SLC6A4, and BDNF, and promote endocrinal, neuronal, and behavioral functions. So meditation, exercise, and nutrition are all known ways to improve your overall epigenome. Unfortunately, there hasn't been any research done on specific ways to improve the epigenome in the human amygdala or hippocampus. But practices that work over the whole body will probably affect those as well. That's all I have for this video. If you enjoyed this, please click the like and subscribe buttons and leave your thoughts in the comments below because it helps the YouTube algorithm. And I'll see you next time on The Sexy Universe.